Today we are going to talk about the receptors. So at the end of this session, you will be able to know the definition of receptors. We will be talking about the classification of receptors. We will be talking about generation of receptor potential. And lastly, we will be talking about the properties of receptors. So these are the 4 things you need to remember regarding the topic of receptors. Now first coming to the definition of receptors. Now receptors are modified nerve endings. Okay, so these are modified specialized nerve endings which are going to transmit the information to the CNS. So receptors, they are going to collect the information from the external environment, plus they also find out things which are happening in the internal environment. So both this information either from the external environment or from the internal environment is carried by the receptors to the CNS. So these receptors, now we know that all the information which is carried to the CNS is in the form of action potential or electrical signals. Now the stimulus can be varied. In the external environment, stimulus can be mechanical stimulus. In the internal environment, it could be changes in your blood O2, CO2 or many other chemical changes, changes in the pH, change in the temperature. So all these varied type of stimuli are carried by the receptor in the form of electrical signal to the CNS. So by or function or definition, we can say receptors act as transducers. So whatever will the type of the stimulus, they convert the varied type of stimulus into the electrical impulses, and these impulses are then conveyed to the CNS. So that is the definition of and function of the receptors. Now coming to the next part is the classification. Now before I move to the classification, one important point I want to tell you is we have defined them as modified specialized sensory nerve ending. Now here you should be careful because we also talk about receptors in term where we are talking about on the cell membrane the ion or a neurotransmitter or a hormone binding to the receptor. So those receptors, slightly different term we are talking about. Here we are talking about are the modified sensory nerve endings. So these are 2 different ways where we are using the term receptor. Okay. Now coming to the classification of receptor. Now there are various ways in which receptors can be classified. So firstly we will classify them according to the type of stimulus. Now according to the type of stimulus we can classify them as mechanoreceptors. So these receptors are going to respond to any mechanical stimuli like touch, pressure or vibration. So first type is a mechanoreceptor. Then we have chemoreceptors. Chemoreceptors, they sense the chemical changes in the blood. For example, they can sense the pH or they can sense the changes in the O2, CO2 levels. So those are the chemoreceptors. Then coming to the thermoreceptors. Thermoreceptors, they can sense changes in the temperature, so it can be hot or cold. So we can have separate receptors for sensing the hot and the cold. Then we have photoreceptors in the retina which respond to changes in light. And then lastly we have the nociceptors which respond to a painful stimuli. So according to the type of stimulus, we have classified them as mechanoreceptors, chemoreceptors, thermoreceptors, photoreceptors and nociceptors. So the other way of classifying is according to the location of the receptors. So this was classified by Sherrington. So according to the location of the receptors, we can classify them as exteroreceptors. So these receptors sense the external environment. So these are cutaneous receptors present in the skin which sense the touch, pressure. Those are exteroreceptors. Then you have enteroreceptors which respond to changes in the internal environment. So these are your chemoreceptors which sense, say for example, the pH or O2 and CO2. Then we have osmoreceptors which sense the osmolality of the body fluids, or glucoreceptors which sense the glucose level in the body. So those are your enteroreceptors. Then we have specialized type of enteroreceptors which are called as proprioceptors. These receptors sense the joint position, the movement of the body. For example, you have the Golgi tendon organ and the muscle spindle. So the muscle spindle senses the muscle length, and the Golgi tendon organ senses the muscle tension. So these are proprioceptors. So this is one way of classification according to the Sherrington: exteroreceptors, enteroreceptors and proprioceptors. Now another lesser known way can be classified anatomically as receptors for special senses. So those which sense the vision or the hearing, smell, taste. Then we have receptors which are cutaneous receptors. Then we have receptors for deep sensations, that is your proprioceptors. These are deep receptors and coming from the viscera, visceral receptors. So they are going to sense the sensation coming from the viscera. Now in this diagram we can see the cutaneous receptors mostly. Now here we have receptors for touch. So for fine touch, you have two different receptors which can sense the touch sensation. This is a capsulated receptor which we can see, which is nerve ending. This is the Meissner's corpuscle which senses the touch. So it is a cutaneous type of receptor. You also have another receptor for touch which is called as a Merkel's disc. So both these receptors are cutaneous receptors and they can sense the touch. Then you have receptor for pain. So the nociceptors, or these are the free nerve endings which sense the pain. These are nociceptors, these are free nerve endings. Then you have receptor, this is a Pacinian corpuscle. When deep pressure is applied, there is stimulation of the Pacinian corpuscle which is present in the dermis. So this is a deep type of receptor present in the dermis. So responds to deep pressure as well as vibration. So deep pressure can also cause sometimes pain and can stimulate the free nerve endings as well. Then you have thermoreceptors which can also be, these are also cutaneous receptors. They can sense the heat and cold. So these are the Rufinus endings which usually respond to warm temperatures. So these are Rufinus endings. So these are different types of cutaneous receptors. So now we have classified the receptors. So we know that the receptor is going to convert different type of stimuli into the electrical impulse. So now what is a further course of action? So we should know what is signal transduction. That is, now the receptor has sensed the stimulus. Now this stimulus has to be carried in the form of electrical impulse to the CNS. So we are going to take this Pacinian corpuscle and zoom it out here, the structure of the Pacinian corpuscle. Now Pacinian corpuscle is a very large receptor and anatomically it is very easy to isolate it and microdissect it. So Pacinian corpuscle is taken to study the production of receptor potential, because receptor potential is going to help transmit the impulse in the form of electrical signal to the CNS. So this is the structure of the Pacinian corpuscle. The Pacinian corpuscle contains this central unmyelinated sensory nerve ending. So this is one part of the Pacinian corpuscle, the central portion of the unmyelinated nerve fiber. This central portion in turn is surrounded by the connective tissue capsule. So you have the concentric lamella of this connective tissue capsule. Now apart from this sensory nerve fiber, this continues as the first node of Ranvier, which is also within the capsule, and beyond that the second node of Ranvier where the nerve will leave the capsule. So this is the structure of the Pacinian corpuscle. Now whatever be we have seen that different types of receptors, we have mechano, chemoreceptor, thermoreceptor, we have the nociceptor, and we also have the photoreceptors. Whatever be the type of stimulus, whether it is a mechanical stretch or whether it is a chemical change or whether it is light is an exception, I will tell you, and we have the temperature, one more, mechanical, chemical, light and temperature. These are the basic four things. Whatever be the type of stimulus, finally what they do is cause change in the membrane permeability. So they change the membrane permeability of the receptor. Now when the membrane permeability of the receptor changes, that is important, and as it is going to generate a receptor potential. So whenever you give a mechanical stimulus, for example you stretch, when you stretch the membrane, it increases the membrane permeability, and when the membrane permeability usually is in the form of a depolarizing current, so depolarization of the membrane occurs and that generates a potential across the receptor membrane, which is called as a receptor potential. Similarly, chemical change will also bring about change in the membrane permeability and cause receptor potential. This is again a depolarizing type of potential. Similarly, change in the temperature changes in membrane permeability, influx of ions, depolarization and receptor potential. So most of the time for mechanical, chemical, temperature, it is a depolarizing potential. Only exception is light, where it is a hyperpolarizing potential. So we can study the detail when you are studying the special senses, but right now mechanical, chemical and temperature changes cause a depolarizing potential, whereas light causing a hyperpolarizing potential. Now we will see how this receptor potential in turn is converted into an action potential. So now we will take the example of the Pacinian Corpuscle, which is a mechanoreceptor. So when a mechanical stimulus is applied here, you apply a stimulus to this concentric lamella. This stimulus is going to cause deformation of this central structure, which is the unmyelinated portion of the axon. Once this deformation occurs, it is going to cause change in the membrane permeability of the ions in this area, and that causes generation of receptor potential. Now if the stimulus strength is enough, this receptor potential, it will propagate towards the first node of Ranvier, and when this potential reaches around 10 millivolt in the first node of Ranvier, it will create local circuits of current flow, and that will lead to production of action potential in the axon, and this will be propagated forward to your CNS as a form of electrical impulse. So we have given a stimulus, so we can just make a flow chart of this. First is arrival of the stimulus. Now here this is a mechanical stimulus here, it could be anything. So your first step, will be your steps in how signal transduction occurs. What do you mean by signal transduction? That is conversion of the stimuli into an electrical energy and information carried to the CNS. That is your signal transduction. So first is arrival of the stimulus, which is, in this case we are taking example of Pacinian Corpuscle. So the mechanical stimulus that will cause deformation of the unmyelinated portion of the nerve fiber, that causes change in the membrane permeability of the membrane permeability of the receptor membrane, causing depolarization. This depolarization is a generation of receptor potential. Okay. Now as you increase the strength of the stimulus, greater will be the amplitude of receptor potential. This point you should remember. So you increase the strength of the stimuli, greater will be the amplitude of the receptor potential. Now once the amplitude, it reaches a sufficient amplitude enough to create a local circuit of current flow in the first node of Ranvier, then that will cause propagation of action potential. So this will cause action potential in the nerve fiber. So this first node of Ranvier acts as a point where the receptor potential is converted into an action potential. So it has to be this spike generator zone, it has to reach more than 10 millivolts for this to fire. So it will convert the generator potential of the receptor into an action potential in the nerve fiber. So this will be converted. So this stimulus is finally converted into an electrical energy and propagated, and the CNS will interpret as a mechanical stimuli. Now an experiment can be done to find out what is the site of generator potential, or where actually is the receptor potential produced. So we have one electrodes are on the unmyelinated nerve ending, and you have another electrode on the node of Ranvier. So first thing, you have given the stimulus and you have seen with the concentric lamella a generator potential can be measured via the CRO. Now when you remove this lamella and directly stimulate the unmyelinated nerve ending, still receptor potential is produced. Secondly, what you can do again, second point, you can block the first node of Ranvier. Now, when the first node of Ranvier was blocked, the receptor potential still could be recorded, but action potential was not recorded. So that means that the receptor potential is generated within this unmyelinated nerve ending. Okay, so this is the site of generation of the receptor potential. So to sum up the properties of receptor potential, it is a graded potential like your EPSP. So on its own it cannot generate an action potential. So we have seen here, this is the node of Ranvier. We have a deformed area, there will be depolarization of this local membrane producing receptor potential. Only when it reaches a sufficient magnitude, then only the node of Ranvier will be stimulated and it will convert the receptor potential into action potential by forming this local circuits of current flow. So it is a graded potential. Then this receptor potential, magnitude of receptor potential depends upon the directly proportional to the strength of the stimulus. Greater the strength of the stimulus, more will be the magnitude of receptor potential. And lastly, the receptor potential magnitude will decide the frequency of firing of action potential. So greater the amplitude of receptor potential, greater will be the frequency of firing of action potential. So that tells you greater the strength of stimulus. So from these two we can derive greater the strength of stimulus, greater will be the firing rate of action potential. So that is how the brain is going to interpret the stimulus strength. So by increasing the stimulus strength, first you are increasing the magnitude of receptor potential, and as the magnitude of receptor potential increases, more will be the firing of action potential. So in that way, strength of the stimulus is increased, your frequency of action potential will be more, and that is how the brain is going to interpret what is the strength of the stimulus. So now we are going to talk about the properties of receptor. So first important property of the receptor is specificity, or also called as a law of adequate stimulus. Now each receptor is specific for the type of stimulus it is going to respond to. For example, the rods and the cones, they respond to light. So they are highly specific as they respond to only light. Then we have receptors for touch. For example, the touch receptors are going to respond only to touch. The Ruffini's endings are highly specific for warm temperature. So this is the property of specificity or law of adequate stimulus, where each receptor is going to respond to only a specific type of stimuli. Next property is the label line principle, or some books are also giving that as Mueller's doctrine of specific nerve energies. Now what is this label line principles? Now we have seen that there are different type of sensations. You have mechanical, chemical, thermal, electrical, so many different type of receptors and sensations, but all these impulses are carried in the form of action potential. Then how does the brain interpret whether it is a touch, or whether it is a pressure, or whether it is a change in the temperature? So how does the brain interpret? So that is decided by this label line principle, which states that each nerve fiber, or each nerve fiber in the tract which is carrying the different sensations, they go to specific areas in the brain. So for example, we know that the impulses from your rods and cones, they go to the visual areas or the visual cortex of the brain. Okay, signals coming from the ear, they go to the auditory area in the brain. Receptors, there you have specific areas for touch. So we know the touch goes to the parietal zone, and different areas, all the areas in the body are mapped in the parietal somatosensory areas. So that is each nerve fiber is going to a specific area in the brain. So that is Mueller's doctrine of specific nerve energy. So wherever along the pathway you stimulate, if it is going to a particular area in the brain will interpret that particular sensation because the area is specific for that sensation. So that is a label line principle, or also called as a Mueller's doctrine of specific nerve energies. The next very important property of the receptor is adaptation. Now we have seen that when you apply a stimulus, receptor potential is generated, and if it is of sufficient magnitude, it will generate an action potential. So as you increase the strength of the stimulus, we saw that it increases the magnitude of receptor potential as well as the frequency of action potential. But if the stimulus strength remains constant, if there a constant strength of the stimuli is there, after some time the action potential frequency goes on decreasing and finally it can stop also. So there will be decay in the generation of action potential. So that is the property of adaptation. When a constant stimulus is given, the action potential initially frequency will rise, but after some time the frequency of action potential goes on decreasing and will completely stop to respond. So with the constant stimulus strength, adaptation of the receptors occurs. Now depending upon, all the receptors may not adapt in the same way. We can have different types of the way in which they adapt. The receptors can be classified as tonic receptors, which are slow adapting or non-adapting receptors. These are slowly adapting receptors, or they can be completely non-adapting receptors. For example, we have the muscle spindle, joint receptors. These are slow adapting, and non-adapting, very important, is your pain receptors. Now the muscle spindle and joint receptors, they continuously appraise the brain about the position of the joint or the position of the body in space. So that is important to maintain your posture. So they are continuously sending information to the brain, so that is important. They do not adapt. And you have the pain receptor. Obviously the pain, as soon, whatever time the pain remains, the signals of pain are being transmitted to the CNS, and this has a protective value. So pain receptors will never adapt. So these are tonic receptors. Then you have the phasic receptors, which are rapidly adapting receptors. For example, you have the Pacinian Corpuscle, which respond to deep pressure and vibration, and your touch receptors. Then olfaction, olfactory receptors. So immediately when you are entering a particular room, if there is a strong smell, you feel the smell. But if you remain in the room for some amount of time, after that the smell is not going to annoy you. So that is because of adaptation of the olfactory receptors. Similarly for touch receptors, we don't feel the touch of the clothes always, isn't it? So this is why, because what is the importance of this phasic receptors is, lot of information going to the CNS has been prevented, unwanted information. So lot of unwanted information going to the CNS can be prevented as they rapidly adapt. So those are phasic receptors. So we have two type of receptors depending upon how they are adapting. You have the tonic receptors, which are slowly adapting. We have the joint capsule receptors, muscle spindle receptors, and pain receptors, which are completely non-adapting, so they have a protective function. And we have the phasic receptors, which are fast adapting. So touch receptors, you have the olfaction, so they prevent unwanted information going to the CNS. So that is how the adaptation can be studied via these two receptors. Now what is the actual mechanism of adaptation, general mechanism of adaptation of receptors? Now firstly, for example, it could be when we take the Pacinian corpuscle. We have studied the structure of Pacinian corpuscle. When you apply the stimulus, we have seen that there is change in the structure of the unmyelinated nerve ending. So when a constant stimulus is applied, there is redistribution of fluid in the structure. So suppose this concentric rings were there, and this is a viscous structure, you applied pressure here. So after some time the fluid within this will readjust and the deformation will no longer be present to stimulate the central nerve ending. So there is redistribution of fluid, and that is a cause of adaptation, especially in the Pacinian corpuscle. Another reason for adaptation is accommodation of the sensory nerve fiber, which is carrying the impulse. Now what happens when this stimulus remains constant? There is inactivation. We know that it is a depolarization of the membrane potential, and then action potential is conducted. So if you give a constant strength of stimulus is remaining there on the membrane of the receptor, it causes inactivation of the sodium channels, and I am sure we know when the sodium channels are inactivated, it no longer can produce the next action potential, or it fails to respond to the next stimulus. So that because of this inactivation of sodium channel, that causes accommodation of the sensory nerve fiber, and that is one way by which most of the receptors are adapting. So this is how adaptation occurs, either via redistribution of fluid or change in the structure of the receptor, or because of the accommodation of the sensory nerve fiber. Next property is the law of projection. So what does this property state? No matter where along the pathway you stimulate, the sensation will be produced at the point where the receptor is present. So what does it state? Anywhere along the pathway you are stimulating, the sensation will be produced at the area where the receptor is present. So that is the law of projection. So this can be best explained with what is a phantom limb, for example. So the best example for law of projection we can take as a phantom limb, or it is a non-existing limb. Here the patient complains of pain in the limb which is not present. The limb is not present. So what is the cause of this? This is because at the site where the patient is feeling the pain, there is irritation of the nociceptors and proprioceptive fibers. And these fibers, because of the irritation, they carry their pain impulses, and the patient perceives them as coming from the limb where the receptors were initially present. So this pain is perceived at the site where the receptors or the organ was initially present. So at the stump of the amputated limb there is irritation, and because of this they carry the nerve signals to the brain, and that is the brain interprets as pain coming from the initial site where the limb was present. So that is the phantom limb. Okay, so we have seen specificity, where each receptor is specific to the type of stimulus it is going to respond to. Then we have seen the label line principle, or Mueller's doctrine of specific nerve energies, which states that wherever or however you stimulate the nerve pathway, there is a specific area in the brain which is stimulated, which will produce a response in the receptor. Then we talked about adaptation. So we had to classify them into tonic adapters and phasic adapters. Tonic were slowly adapting, phasic which were fast adapting receptors. We saw the significance of adaptation. When they are phasic adapters, they don't want unwanted information going to the brain, and for tonic receptors, they want the brain to be continuously appraised of the damaging stimuli. Then we talk of law of projection. So wherever what is the site of the receptor, there the sensation will be felt. That is, we have given the example of phantom limb. Now coming to our last property, the law of intensity discrimination. Now how does the brain interpret whatever the stimulus is coming, whether that stimulus is weak, moderate or strong? So there are two ways by which the brain interprets the intensity of signal. One is by the frequency of action potential. So we have seen greater the stimulus strength, greater will be the frequency of action potential. So depending upon that, the brain can interpret whether it is weak, moderate or strong. So if it is a weak stimulus, the number of action potential fired will be less compared to moderate and a strong stimulus. So that is one way. Another way of sensing the intensity is whether it is weak, moderate or strong is the recruitment of motor unit, recruitment of the units, sensory units, in this case. Now what is a sensory unit? Sensory unit will be the area which the receptor supplies to the skin. For example, here there are many nerve fibers which are innervating this particular skin. So each nerve fiber with the area it innervates, that is called as a receptive field for that receptor, for that nerve fiber. So we have one nerve fiber which will innervate a particular area. So it is going to branch out and innervate one particular area. So different nerve fibers will have overlapping receptive fields. Now we can just zoom out here and see, when a weak stimulus is applied, only some of the sensory units will be recruited and it will fire. So usually the central area, the nerve fibers which have a lower threshold will fire first. As you increase the stimulus strength, more and more areas of the sensory unit will fire, and more number of nerve fibers in the center as well as in the periphery will fire. So those nerve fibers which have a higher threshold are also going to get added up as you increase the stimulus strength. So this is how, by recruiting the sensory units, the brain is going to interpret the intensity of the stimulus, whether it is weak, moderate or strong. So this, by increasing the frequency of action potential, you can discriminate the intensity. This is a type of temporal summation. And by increasing the recruitment of sensory unit, as you increase the stimulus strength, that is a type of spatial summation. So at low intensity of stimulus, low threshold fibers will be stimulated. As you increase the intensity, more number of sensory units will be added up, and that can help in deciding what is the intensity of the stimulus. So that is a law of intensity discrimination. So today we have studied the definition of receptors, we classified the receptors, we talked about receptor potential and the properties of receptor. I hope you have understood the concept. You can please like this video and comment in the section if anything is you feel you need to be added to this. Thank you so much.